Just so you know, the microphone is this box, so like hovering in this area.
So not this, but
Surprisingly, not the one that looks like
right.
That's the box.
I think it's
Set up and then we'll get some.
All right, everybody, let's go ahead and get started while we wait for any last minute people to come in. We minimize. So today we have our first PhD student proposal. We have Landon Teer here with us today. How long have you been in the department? When did you start?
It's my third year right now.
So he's
Since
in his
2021.
third year since 2021. He has been with Dr. Chen, working on good things with glioblastoma. And he's come here to talk to us today about his PhD proposal that he just recently got done with. He's going to talk to you about what he's done and where he's going. So let's give him the floor.
All right, well, thank you, Nick, for the introduction, and hello, everybody. Glad to be here and talk about my work. So, as Nick said, I work with Dr. Chen, and our primary focus is on glioblastoma. And so, without further ado, let's just jump into things. All right, so glioblastoma, which I'll be calling GBM throughout this presentation, is just a form of cancer. It's a primary brain cancer, accounting for about 50% of all tumors. And what we know about cancer is that it's a really terrible disease caused by this uncontrollable proliferation and division of cells. They essentially take up all the space and the nutrients, killing out healthy cells. According to the World Health Organization, as of 2020, about 1 in 6 deaths were attributable to cancer as a whole. So GBM will be making up some small subset of that. But of these cancer deaths, GBM is an especially deadly type of cancer. It has a median survival of only around 15 months from the point of diagnosis, with a 5-year survival of typically less than 2%. So by the time you actually go to feeling symptoms, which can include things such as headaches, dizziness, minor symptoms, within a week, a month, you're already developing full-blown paralysis, organ shutdown, things that can lead to this death in a really quick manner. And this top figure on the right here that I've got is a updated figure of what we call the hallmarks of cancer. You can see we've got this sustained proliferative signaling, we've got changes in cell metabolism, we've got this epigenetic reprogramming. These are all just synonymous with cancer and the disease as it progresses. But for GBM, it's actually been identified that really there are 2 that have kind of emerged as being kind of at the forefront of these hallmarks, which is this avoiding immune destruction and this invasion/metastasis kind of process. And so we would say that GBM is a highly invasive type of cancer, and it's a highly immunosuppressive type of cancer. And I'll be elaborating on these terms as I go. And so when I talk about invasion, in the broad sense, cancer will metastasize and spread from one organ to the other. Now, GBM doesn't really metastasize to other organs, but it still likes to readily spread throughout the brain, which you can see here in this figure, how you'll have a tumor and then this rapid dissemination of these single cells to go on to form secondary tumors. This is a real issue with our current therapies, resection, chemotherapy, radiotherapy, because these are all very targeted means to try and eliminate the cancer. And so when you have cells that are rapidly leaving that site and going on to form secondary tumors, you can't just keep cutting out brain tissue or applying harmful toxic agents to large areas of the brain without risking permanent damage. And so this has really led to the development of these new types of therapies that are a little bit less involved on chemotherapy, radiotherapy, and more focusing on alternatives that are already in the body, including the immune system. And so I mentioned earlier that GBM is a very immunosuppressive type of cancer. And you'll be hearing me use the terms immunosuppression and immunoevasion, just referring to a literal stopping of immune cells from killing the cancer cells, or just an evasion of the mechanisms they use. And so I've included this definition just where immune evasion is, again, the literal avoiding of the immune cells so that they can continue to reproduce, divide, and form these secondary tumors. When talking about the cancer's susceptibility to these immune responses, we'll classically call them as being either hot tumors or cold tumors, with hot tumors being types of tumors that are more susceptible to immune cells and their responses. You can see on the right here how we have a large number of these T cells and natural killer cells that have infiltrated this tumor. GBM, unfortunately, is not one of these tumors. It's a cold type of tumor, which is characterized as having much fewer cell infiltrates and thus less cancer elimination. And so there's been a lot of work into really trying to figure out what are these extrinsic factors that might be leading to this difficulty with immune cells trying to infiltrate this tumor and eliminate them, or these extrinsic factors that are leading to these cells rapidly leaving their primary tumor site to go form secondary tumors. And so in trying to figure out what these factors are, there's been a lot of research into what we call the tumor microenvironment. So this is going to represent the area including the tumor and also surrounding it with all of the vasculature and cytokines and chemokines and so forth, with the soluble agents being what we would call the biomolecular compartment of it. This is going to be your traditional biology when you think of, again, just growth factors and things like that. But in the last few decades, we've seen the emergence of study on this other compartment, which is known as the biophysical compartment. So rather than having these secreted agents that can be interpreted by cells, we now have the physical stimuli of that environment, which is now leading to changes in the cells. These can include the ECM, extracellular matrix, composition, ultrastructure, fluid flow and pressure from that. As you can see in this image on the right, this tumor microenvironment is a very complex type of environment with a multitude of different cells, ECM, which all kind of work to help promote this tumor growth. And so this has only emerged in the last few decades, which means that our findings thus far have really been limited, and it's still not very well understood how these physical stimuli are really leading and contributing to disease progression. And so the biophysical aspects that I've kind of chosen to look at are really referencing the tissue density and ultrastructure, or just the organization of it, and then the stiffness of the tissue. We can see here that when you have normal brain tissue versus GBM type brain tissue, these are two very different types of environments with various levels of ECM and other factors that can be leading to pathway activation. So when you get these different signals that are interacting with the cells, you get varied levels of this pathway activation, which leads to differences in these downstream biological effects, including cell speed, in which we can see when just varying substrate rigidity. So just varying what the stiffness of what these cells are seated on, we can directly control how fast the cells are migrating across the surface. So this is, again, looking into that invasive type question now. And so when you get these cell differences and they go to spread out and form these different parts of the tumors, you get a lot of what we call tumor heterogeneity. So just these different distinct regions of the tumor. And continuing off on the stiffness aspect, we've actually looked at what we call the core and then that invasive edge of the tumor, and found that there are some pretty striking differences in the mechanical properties using the Young's modulus, or just the stiffness, as I've been alluding to. And so with some of this preliminary data and kind of lack of understanding of how these biophysical stimuli lead to disease progression, we really wanted to try and figure out, well, why is GBM so deadly and able to go through this progression at such an accelerated rate? And so my proposal was to investigate these biophysical regulators that are contributing, again, on this invasion front and this immunosuppression front. I'm going to do this by looking through, again, this dense tissue ultrastructure and then these different stiff regions within the tumors. I'll discuss these specific aims a little bit more detail as I get through them exactly, but all of this is for the express purpose to just advance our understanding of this biophysical TME so that we can increase our understanding of GBM progression in relation to these biophysical aspects to potentially develop better therapies and hopefully try and increase that survival time from that 15-month median that we're currently seeing. And so this first part of my data is so far what I've completed in my time here, and it's going to be focusing much more on this invasion and this dense tissue ultrastructure. And so, as I've already mentioned, GBM is a very invasive type of cancer. Spreads throughout the brain, and this is really interesting because if you consider what the brain looks like with the millions of different cells and connective networks between them, such as those shown in this image here, you get a very dense microenvironment with pore sizes that are on the nanometer scale. This is interesting because our cells are over 1,000 times bigger. They're on the micron level scale. And yet GBM is somehow able to go through these pores that are much smaller than them and spread throughout the brain. And this doesn't intuitively make a lot of sense. One of the big reasons why GBM therapies have been so limited is because our current therapies are often limited by these pore sizes, but somehow these cells are going through. And so I really wanted to come up with an explanation for why GBM is able to navigate this level of constraint so easily. And so I wanted to look into these specific GBM types, and ultimately I came up with this first specific aim where I wanted to identify the underlying biophysical mechanisms of PMT that contribute to heightened invasive potential in GBM. And so when I mention PMT, I'm referring to this proneural to mesenchymal transition process. Proneural and mesenchymal are just different phenotypes of GBM in which our proneural cells are less therapy resistant and less invasive, meaning that this would be a more typically early stage of cancer and one in which you would have a better prognosis upon receiving treatment. Whereas mesenchymal GBM is now representing the other end of this axis, where these are highly invasive, going to spread all throughout the brain, and these are going to be much more resistant to those therapies that I've talked about. And so I mentioned that this is a proneural to mesenchymal transition. This can occur because these phenotypes, while distinct, exist on an axis. And with proper stimuli, they're able to transform from this less aggressive proneural state to the more aggressive mesenchymal state. This is inducible by, again, a number of signals, one of which is TGF-beta. This is just a common cytokine in the body involved in a multitude of processes, but ultimately, in this context, it contributes to a more pro-tumor type environment. There have also been some studies to suggest it has a little bit of an immunosuppressive capacity beyond just inducing this PMT, but we're really focusing on the invasion aspect in this go. And so in an attempt to try and explain why GBM is spreading throughout this really dense environment, I came up with the hypothesis that perhaps the cells are seeing a reduction in their stiffnesses so that they can better deform their bodies to traverse this level of confinement. I love to use the example of you can have your round hole with your square peg, and it doesn't normally try and fit through. But what if instead of say, a wooden square peg trying to go through a hole, we now change that to a wet clay. And now it's able to actually change its shape to go through. Perhaps a simple explanation, but one that we thought might be interesting to look at. And so I employed the use of many traditional biological assays, as well as some engineering platforms that we've designed to really investigate this heightened invasion. The most important, perhaps, being the use of our microchannel devices. So the brain is, again, a very dense, confined environment that these cells are constantly having contact on all axes. And so to try and recapitulate this, we developed these microchannel platforms in which we have channels that vary in size from 50 microns or a more free type space all the way down to 5 microns, which would simulate a more confined space. And so the idea is that we treat our cells, we check their mechanical properties, put them in these channels and see how that confinement does change. And so the first part of my experiment was to look at, can I actually induce a disease progression type event in these cells, making them acquire that more mesenchymal phenotype? And so in our lab, I used 2 types of cells, U87 and U251. These are both human derived cancer cells, with U87 being a cell that is classically defined as a more mesenchymal type cell already, with U251 being more on the proneural side of that axis. And so using TGF-beta that I mentioned earlier is well categorized as inducing this event. I applied it to my cells for 3 days, and using Western blots, I was able to verify that TGF-beta receptor binding was occurring as seen by this upregulation in the phosphorylation activity of the SMAD2 pathway. This is the canonical pathway for TGF-beta signaling, and thus would be expected to increase with the addition of TGF-beta. I then looked at the phosphorylation activity of the STAT3 pathway. This is a almost intermediary pathway in a mesenchymal transition in which cells that are beginning to see mesenchymal pathways activated will begin to upregulate this activity for phosphorylation of STAT3. Once again, we see in both cell lines, there was a significant increase. And finally, I looked at ZEB1. This is a well-known mesenchymal transcription factor indicating the cells have now gone through that mesenchymal pathway and are adopting that mesenchymal phenotype. Once more, we see in both cell lines a significant increase, indicating that with my TGF-beta addition, I was successfully able to induce this PMT process in my cells. And so after inducing this PMT, I was then curious, are there protein level changes now that correspond with this event? And so I looked at the lamin and the actins, as these are the primary structural proteins in the nucleus and cytoskeleton. This felt as the easiest way to divide up the cell to see how these regional effects may be occurring. And so we see for U87 and U251 that most of the proteins exhibited a significant decrease in expression, with U251s showing the general trend of decreasing in their lamin expression. All this to say that the total amount of these structural proteins has now decreased following this PMT. With these lower expression levels, I also noticed that there were these organizational differences when looking at the cells, where you can see in my top box here for U87, there's a lot of F-actin or filamentous actin. This is going to be the much more rigid form of actin. Yet after TGF-beta, you see it's not nearly as prominent as it was. Moreover, in the U251s, again, we see a little bit of this F-actin spreading throughout the entirety of the cell body. Yet when we go to look after TGF-beta, it's been reduced again. With the lamin profiles and looking at the nucleus, we observed that there was an increasingly dysregular nuclei shape following TGF beta expression, or sorry, my computer turned off, TGF beta addition. And so we see that these actins are now being expressed at a lower level and not being expressed throughout the cell, changing in where they are localized. We see the lamins are being expressed at lower levels, and now the nuclei are taking on these increasingly irregular shapes. Additionally, when zooming in on that nuclear envelope, we see that there were these pore-like structures that were beginning to form after TGF beta, which intuitively makes sense with a lower expression, and then also helps to corroborate this difference in organization of these proteins. All of this is to say that we are now getting these protein level changes with this induction to that mesenchymal aggressive phenotype. And so after seeing these protein level changes, I would then need to verify, are the mechanical properties actually changed in these cells? And so to quantify this, I used atomic force microscopy. In short, you have a laser that is shining at the end of this cantilever with this tip and reflecting back onto a photodetector. As the tip rasters across the surface of my sample, my cells in this case, there's going to be a deflection. And using the known stiffnesses and deflections of these lasers, I can then back calculate the stiffnesses of my cells. And so in looking at my U87 and U251, you'll notice that for the non-treated cells, the range of moduli or stiffnesses of these cells is pretty spread, especially in the U251. But once I add TGF beta, we see that these ranges begin to consolidate into a much narrower region. And this region is on a lower Pascal reading, with my average moduli for my U87 going from about 900 and 400 Pascals down to about 450 and about 350 Pascals. My U251's going from almost 2 kilopascals in the cytoskeleton and about 1200 Pascals in the nucleus down to 1200 in the cytoskeleton and about 600 in the nucleus. These are some very large changes that are accompanying this TGF beta treatment. And with this reduced Young's modulus, I'm now demonstrating that, yes, the cells are becoming softer now. And interestingly, we saw that while the cytoskeleton was significantly reduced to a much greater level across both cell lines, the nucleus was only reduced at a very great level in the U251s, the U87, which was much less pronounced. And so I got to thinking, after looking at this, well, considering the difference in the shapes in my nuclei and the formation of these pore structures, considering that in the more proneural line that is perhaps more susceptible to this PMT process, having the greater changes in both regions, whereas the already mesenchymal line isn't experiencing as great of changes in the nucleus, perhaps that could be a good place to start on trying to figure out, is this a limiting factor for how my cells are moving? And so after looking at this reduction in mechanics, I then had to move on to the next step of seeing, does this actually impact the invasiveness that I'm trying to study in these cells? And so I mentioned my microchannel devices earlier. I've included a couple of videos, with my left video being the 50 micron size channels. As you can see, the cells are able to not interact with the walls as much. They can migrate freely throughout. Several of them like to go over the channels, and I don't really like that. But for the most part, you can see that they have a lot of open space to just go through. Whereas in my 5 micron channels on the right, you can now see my cells are much more confined in the walls. They don't have that free motility to oscillate around their environment and seem to have a much more directed type of movement. So after applying my TGF beta, which I know reduces the mechanics, I wanted to see, does this reduction correlate with some change in the behavior of these cells moving through the channels? And what we saw was that in the U251, there was a significant increase in cell speed in these tightly confined channels, as well as in the others, indicating that the cell is typically going to have a more invasive type of phenotype. A little surprisingly, we saw the U87s did not share this trend. But again, you have to remember that this U87 is already a more mesenchymal type cell. And so I hypothesize that the effects of TGF-beta in inducing this PMT and this more invasive phenotype are simply less pronounced, or perhaps beginning to have a negative effect on the U87. But nonetheless, we showed that for the less aggressive cells, which would typically begin the tumor and develop into a more aggressive and deadly type, we see this increase in confinement with this reduction of mechanics. And so the next step, or the final step of this AIM-1, is to then see, can I reverse this trend? And so as I mentioned, these cells are getting softer with TGF-beta. And so I thought perhaps by stiffening the cells, I could then limit that acquired invasiveness. And so I used a nuclear stiffening drug and applied it to my cells after they've been exposed to TGF-beta. We see that for U87 and for U251, cell speeds always returned to levels no greater than experienced by cells that had never been treated with TGF-beta, which is to simply say that they got slower even after they had that heightened invasiveness. And you can see this in the video, whereas previously I had cells that were migrating very quickly through, and now you see they're only kind of shaking in place now. They can no longer freely migrate through those confined environments. And so with that final experiment, I then get to the end of my first aim, which, just to summarize, was that by using TGF-beta, I was able to induce this PMT event in my GBM. When the cells acquired the mesenchymal phenotype, they experienced lower expressions and then organizational differences of their structural proteins that correlated with a reduction in their stiffnesses. This allowed them to better migrate through these confined environments, which was then subsequently reversed by stiffening the cells back up. Looking ahead, I would potentially want to look at other pathways and mechanisms that could be involved in this cell stiffness and that change. I would also want to look at 3D type environments, as these in vitro 2Ds, while better in that they are showing that confinement aspect, are still not a perfect representation of the brain. And then, of course, as I've kind of alluded to earlier, I would also want to look at this immunosuppression type aspect. Which brings me on to my AIM-2, which is less far along than I would like, but currently in the works. So in looking at this differential stiffness across the cells, I would really want to, as I mentioned in the last part, try and increase that recapitulation or that biomimicry aspect in order to get a more accurate response of how my cells would interact. And so to know what this environment is and how to recapitulate it, we need to consider what are these primary bindings that are occurring with GBM. And so we see that HA, or hyaluronic acid, is going to be your primary ECM component comprising all of that tissue, with CD44 being the binding receptor that is overly expressed on GBM. This binding and pathway activation corresponds with pro-tumorigenic behavior, including an upregulation in angiogenesis, these metabolic shifts, and then importantly, this cancer cell invasion that I studied in AIM-1. And so this axis represents a very powerful tool in trying to bridge the gap between this 2D and now this in vivo type response that we would expect to see. And this is really because HA is a very dynamic type of ECM. Depending on the form it's in, whether it's high molecular weight or low molecular weight, you can either get a homeostatic type response or what you would normally expect of healthy brain tissue versus a pathophysiological response, or one that we would expect more with cancerous tissue. Excuse me. And so by introducing now this HA component, again, we are starting to recapitulate those in vivo results, which is important because HA binding has also been implicated in this EMT and PMT processes that are very distinguished of cancer. And so after identifying these as being the most likely binding interactions to be occurring, I wanted to see, does HA and CD44 have some effect on the mechanosignaling of GBM? Is it related with this mesenchymal transition phenomenon that I explored in AIM-1? And does it contribute any to what I call immunomodulation, or just, again, relating to evading of this immune system? And so we know that GBM is surrounded by a wide variety of immunosuppressive mediators, in which we see that when taking cells and using this IGF-BP2, just another means to induce mesenchymal transition, we see that there was a significant reduction in the total amount of T cells or immune cells that would typically eliminate GBM as compared to those in which it did not have this transition occurring. And so with this mesenchymal GBM, I explored the STAT and ZEB effectors in AIM-1. And interestingly, these are downstream of CD44. Again, that primary binding axis of the ECM of the brain with these cancer cells. And this axis and these effectors are also associated with the production of this IL-6 and IL-17, these pro-inflammatory cytokines that are going to have the double effect of, one, promoting just tumor development, but two, inhibiting immune cell activity that could potentially eliminate the cancerous cells. And so it follows that after seeing ZEB and STAT activity are related to the CD44, and knowing that CD44 binds with HA, I began to wonder, is there some kind of mediated production of these pro-inflammatory cytokines that help develop GBM? And so I had the hypothesis that stiffer gels will lead to greater binding, which will ultimately result in a greater production of these IL-6, IL-17 to then create a more immunosuppressive environment. And so for this aim, the main goal is, of course, to generate these HA platforms that I've been alluding to thus far. HA is a very useful material because it's very easily modified. In particular, I talk about these tumors having different stiffnesses across the different regions, and the brain tissue changing in stiffness as more GBM enters a localized area. And so to try and figure out how these different GBM are responding in these different environments, I would want to be able to tune my gel into these different stiffnesses. And so this is accomplished by taking the HA molecule here and using a cross-linker. We use DTT, which essentially takes these strands and helps increase the amount of binding that can occur. And with this binding, you then have smaller pores and a more rigid type substrate, thus leading to greater stiffnesses. I would end up repeating some of my previous biological assays and then really begin to interrogate, is this CD44 binding really important in the STAT3 ZEB1 expression for my GBM, and how does this ultimately contribute to those secretion profiles of the interleukins 6 and 17? And so we consulted the literature and found that we can tune our HA gels according to the weight percent of HA that we add and our thiol repeating units. And again, in creating this cross-linked HA, we saw that previously with PA gels, with proneural and mesenchymal type cells, we were not able to quantify a difference between the invasion profiles of them, which is to say that we couldn't see how they were changing and moving very well, as evident in this first image here. But when we begin to use HA-based materials, we can now tease out these differences where you'll see the mesenchymal cells are exhibiting that increased behavior of spreading out from that initial tumor site. Moreover, we see that the method with which we present the HA is again very important in determining how these cells behave, with the low molecular weight, again being that more pathophysiological type response, HA being more indicative of regular brain tissue. We see that there are significant differences in these presentations versus a traditional PA gel type platform. But most interestingly, we see when you add in RGD, you begin to add in now an integrin-based mechanotransduction binding. And with it having the greatest speed, we see that this binding interaction is very important in determining how the cells are able to move throughout their environment. And so as of this experiment, I still have to verify the HA weight percent scale. By that, I mean we're using these values, but we ultimately want to ensure that we are at least in the same general ballpark of these modulus values. I then need to culture on my HA gels, perform my knockouts to look at this binding axis, see how my downstream effectors are impacted by the loss of that sensing. So my expected findings would be that the mechanosensing of this axis does regulate these mesenchymal transitions. We see that through the increase of STAT3 and ZEB1, similar to that of my findings in AIM-1. GBM on stiffer HA gels will have the greater production of this IL-6, IL-17, thus making a more immunosuppressive type environment. And so looking beyond this step of the project, I would really want to see how do these changes in the production of these pro-tumor anti-immune cell interleukins, how does that really impact these immune cell populations that may be present in the brain environment? Which brings me to my final aim, which would really be looking at, again, how the immune cells are now directly impacted as a result of this HA-CD44 axis. And so immune cells, as I've been talking throughout my presentation, are going to typically have an anti-tumor type effect. Some will have a pro-tumor effect, and it just all comes down to the types of cytokines they produce, the amount of binding they're able to achieve, and also how they interact with other immune cells. There's a cross-regulation where certain immune cells can actually inhibit the activity of anti-tumor cells in order to lead to a more pro-tumorigenic type environment. One such cell is the monocytic myeloid-derived suppressor cell, or the MMDSC. These are known to inhibit T cell function, in which you can see here when we have T cells that are just naive or unactivated, they have a very low level of proliferation, which is to be expected. When exposed to an activating bead, simulating a binding to their target they want to eliminate, we see there's a significant increase in the amount of proliferation. But upon the introduction of now these MMDSCs, we see that that amount of proliferation has now returned back to that baseline level, indicating that they have a very potent inhibitory effect on the immune cells that would otherwise try to eliminate GBM. And so you might be wondering why I'm talking so much about the MMDSCs in particular, and that's because they actually have a very interesting relationship with GBM. And so in this aim, I would really want to, again, dissect that axis, HA and CD44, seeing are there any downstream effects on this MMDSC population, and thus the amount of reduced activity on eliminating cells such as T cells. And so I mentioned that MMDSCs have this relationship with GBM, where we see that MMDSCs, when in the presence of GBM, are able to develop these, the progenitor cells are able to develop this more MMDSC type phenotype. And in healthy patients versus glioblastoma patients, we see that there is a significant difference in the amount of MMDSCs in that environment, which would suggest that there is a more inhibitory effect of this immune-mediated cancer elimination. And so, considering that we know these pathways produce these immunosuppressive cytokines, and knowing that these MMDSCs naturally are found in high quantities with our GBM, it kind of brings to question how or why are there these MMDSCs in our GBM? Could there be a type of signaling response recruiting them to the tumors? Is it based on the stiffness or the composition of the ECM? Is this a process that accompanies the PMT that I've described previously? And so I generated the hypothesis that yes, there is a response, and the response is that these HA-rich regions in the GBM tumors have an increased presence of these inhibitory immune cells, which leads to a reduction in T cell activity. And it's all due to the CD44 HA binding axis that I explored in AIM 2. And so I would then again repeat my HA-based platform cold culture experiments and add in supernatant of my GBM cells to these primary monocytes just to confirm the differentiation of these MMDSCs. And then I would want to look at the transwell assays in order to determine, does this supernatant or do these GBM cells actually have some kind of signaling effect and recruit these MMDSCs to their location? I would then want to look, perhaps most importantly, at this in a more in vivo type environment using mouse xenografts to see how this interacts now, not on my in vitro environment, but again, in this biologically more accurate type system. After implanting the mice with the tumors, sacrificing them and harvesting, I would want to use mass cytometry to identify if these HA regions do follow my hypothesis and have this high MMDSC and low T cell population. I use mass cytometry because while similar to flow, it has the added benefit of using these metal ion isotopes with a much more distinct spectral visualization, which would allow me to look at a wider variety of targets at once. And so kickstarting some of this aim is a little bit of preliminary data we have in which we had, again, these donor cells that are progenitors to the MMDSCs. And we see that when cultured in the presence of U87, our mesenchymal GBM, you see a noticeable population shift with a downregulation of HLA-DR, which is a commonly used metric for identifying immune cells via their binding potential. If immune cells, such as T cells, cannot bind to their targets, they are not able to eliminate them. And so with this reduction of HLA-DR, you're getting to that naive or that more immunosuppressive MMDSC population with about a 30% change, as according to B. Furthermore, we see that when looking at these proneural versus mesenchymal type cells and adding them with these MMDSCs, we see that there is a noticeable difference in the amounts that express this same profile, with mesenchymal introducing about 15 to 20% more cells of that inhibitory phenotype. This has led then to a reduction in the T cell proliferation when cultured with greater quantities of MMDSCs, thus giving a little bit of support to my different steps as I go along. But again, this is just the preliminary data on it. I still have to, again, culture on my HA platforms that I'm developing, use those trans wells to see is that recruitment aspect there, and then finally do my xenograft model so I can acquire the tumors and begin analyzing these different regions of the tumor for these different immune cell populations. And so that's just expected findings, which are, of course, in the positive at this point until I'm shown otherwise, but that'll take me to the end. I'd like to thank the Speed School, our department, for the funding and the support that I've received, the Chen Lab, some of our funding sources, and that'll bring me to the end.
Do you have any questions for Landon?
So I know you've mostly been focusing on the two cell lines, one a bit more mesenchymal than the other. Do you think you'll add a third cell line, maybe one that's not very motive at all, or one that's like mobile to test your findings once you do a little bit more exploration?
Yes, we do have other cell lines in the lab. We wanted to start off simply just kind of targeting one side of the axis versus the other. I believe we have 2, if not 3 other cells that will fall on different locations of that axis. And so depending on our results, if maybe they're not just as clear as we're expecting, we can absolutely add more, either to clear up those findings or just increase the significance of our findings by adding that intermediary. Yes.
How would you use your findings in this presentation to make better therapeutic?
Yep, so good question. So in relation to aim 1, I'm showing that the stiffening of the cells is reducing that invasive potential. So in that regard, just by applying something as simple as this drug that we've acquired, you can reduce the amount of cells that are leaving the tumor, which is going to increase the efficacy of the standard therapies that are currently being employed. Now, we have to take that with a grain of salt, though, because these standard therapies, while still currently used, are beginning to be edged out by some of these more novel immuno-based therapies. And so that's the reason I have aims 2 and 3 focusing on this immune type population, because by identifying potential axes that are involved in creating this immunosuppressive environment, we can use immunotherapies such as CAR T based therapies, which have a reduction in the amount of receptors that bind and maybe can't now be targeted by these inhibitory signals, and then lead to a cell death of the cancer.
Thank you, Landon.
I guess make sure there's no.
Everybody, thank you for coming today. Next week, in this room, I believe is Dr. Copa. Innovation Garage on campus.
Do you want me just to leave?
Yes, thank you.